This video will focus on RNA transcription and processing, and some of the main concepts we'll focus on are listed up on the screen. And so let's begin our discussion with just a very brief review of the central dogma of molecular biology. This should be review for you. You have certainly seen something very similar to this at some point before in the past. So we're looking at the central dogma here. Essentially, it just refers to this flow of information from DNA, which can replicate itself, and that's what this arrow here represents to the left. So DNA used to create a piece of RNA, which is then used to be translated into protein. So again, just this one-way flow of information from DNA to RNA to protein. So just keep that in mind as we discuss this chapter and the next few chapters. And today we're going to be focusing on what's going on here during the process of transcription, when RNA is produced from DNA. So let's discuss just some very general features of transcription. And so imagine a piece of DNA like we have shown down toward the bottom in blue, so double-stranded, complementary base pairing, 5', 3', and so forth. And let's say one of those strands, namely the template strand, is used to create a piece of messenger RNA. And that's what we see toward the bottom in red. So notice the template strand is complementary to the RNA, and of course the only difference is the RNA contains U's instead of T's. And so just some general features of transcription that I want to point out at this time. So again, first of all, RNA synthesis is based on complementary base pairing. Also, when RNA is produced, RNA polymerase is the enzyme that comes along and produces RNA from the DNA template strand. RNA synthesis occurs in a 5' to 3' direction, just like we saw earlier with DNA replication. And also I just want to point out that for a specific gene, for any gene, only one of the two strands serves as a template strand. However, keep in mind that for different genes, different strands could be used as the template strand, and that's illustrated in the bottom figure here. So we have two separate genes being expressed. Toward the left, we have the bottom strand being used as the template strand. And again, it's used to create a piece of messenger RNA, shown in red. Toward the right, we see the top strand being used as the template strand. So the point is, within a given gene, only one strand is used. However, keep in mind that between different genes, we could have either of the two strands being used as the template strand. A few comments about RNA polymerase. Again, RNA polymerase is the enzyme that actually synthesizes RNA during the process of transcription. I just want to briefly point out that prokaryotes, namely bacteria and archaea, they just have one RNA polymerase, whereas eukaryotes have 3. And so RNA polymerase 1, this is the enzyme used to produce ribosomal RNA. RNA polymerase 2, this one produces messenger RNA. And RNA polymerase 3 synthesizes transfer RNA, as well as small nuclear RNAs. We'll come back to those later. And recall that tRNA, these are the molecules involved in bringing amino acids into ribosomes during the process of translation. So today we're going to focus primarily on RNA polymerase 2, because this is the one that, again, synthesizes messenger RNA. So before we actually look at the stages of transcription, let's just take a brief overview look at what a gene might look like. So we're just identifying some of the major regions that we commonly find within eukaryotic genes. So let's just begin here in this yellow and orange region. This is all part of the so-called coding region. And so this is the portion that will actually be transcribed and used to produce RNA. And notice we identify exons and introns within that coding region. So exons, these are simply the regions that will be incorporated into the fully functional RNA after this gene is expressed. The introns, however, these are portions that will eventually be spliced out. And so introns, unless there are mistakes occurring, introns tend not to be found within fully functional RNAs. But the exons eventually get spliced together, and we'll come back to that briefly. We can also find promoter regions, which tend to be just upstream of the coding region. The promoter region, that's where RNA polymerase as well as other proteins bind in order to initiate transcription. We might find these other regions, for example, promoter proximal elements, and these are just other regions. So we're looking at DNA here. So these are just other regions of DNA at which different proteins can bind to enhance transcription. We might also find these regions labeled as enhancers. And enhancers can be found pretty much all over the place. These are regions that specific proteins can bind here, and again, that can enhance transcription. Likewise, although there are none labeled here, there are also regions referred to as silencers. And so these are just regions can be found pretty much anywhere, just like enhancers. And again, certain proteins can bind there, and that tends to inhibit transcription of the gene in question. So now let's quickly review the stages of transcription. So first of all, initiation. Initiation occurs when various proteins and RNA polymerase all come together and bind at the promoter region of a gene. Once everything is in place, then RNA polymerase, shown toward the bottom as these red circles, RNA polymerase can then make its way down the piece of DNA, and during the second phase, elongation, RNA is synthesized, so RNA bases are incorporated, again complementary to the template strand, again in a 5' to 3' direction. And then eventually, when certain sequences are reached, termination occurs. These are just specific sequences that trigger the RNA polymerase to release from the DNA template, thus finishing transcription. So let's just say a few more comments about each of these stages, specifically initiation. I just want to point out that in eukaryotes and the promoter region, there's a very common sequence, TA. So often that's referred to as the TATA box. So the TATA box, again, is just part of a promoter region. And there's a protein that can recognize that sequence and bind there, namely the TBP or TATA box binding protein. So when the TBP binds to that TATA region, that then recruits other proteins, namely transcription factors. And your book refers to them as general transcription factors, and that's fine. You might also come across the term basal transcription factors, so they both refer to the same thing. But then once all those proteins are in place, that then allows RNA polymerase II, shown here as a red oval, to bind to the promoter region, and then transcription can begin. So once everything is in place, RNA polymerase does its thing, an elongation ensues, and again, that's simply where new RNA bases are added in a 5' to 3' direction, and that's shown here in yellow. And then again, eventually, when certain sequences are reached, termination occurs. And again, essentially, that triggers the release of the RNA polymerase, shown here in green, from the DNA molecule. And there, at that point then, the mRNA, messenger RNA is released. So initially, the messenger RNA is not mature, it's not fully functional. So we say transcription produces a pre-mRNA. Then how is it modified before it's translated? Well, just a few things I want to point out here. So toward the bottom, we see a few figures. If we take a look at this first panel here, notice there is a so-called 5' cap. This just consists of a modified base with some phosphates that's added to the 5' region of the pre-mRNA. And on the other side, the 3' region, there's an enzyme that adds lots of A's. So we have this poly-A tail, so lots of adenines added there. So this 5' cap and the 3' poly-A tail, both of these help to protect the mRNA from being broken down by enzymes. They help to target this as a molecule that can be used for translation, recognized by ribosomes later on. And also the poly-A tail helps to target this as a molecule that can be exported from the nucleus. Recall, of course, transcription occurs inside the nucleus, but then the mRNA has to make its way out of the nucleus into the cytosol, where it can interact with ribosomes during translation. The other thing I'll mention, number 2, before translation occurs, we have to remove the introns and splice together the exons. Okay, so let's discuss that just a bit. So first of all, let's go back to our central dogma figure. Again, we saw this before, DNA to RNA to protein. But what's interesting, if you look at humans, we have 21,000 or so genes, but we can produce lots more proteins. So the question is, well, how can so few genes code for so many proteins? Well, it turns out that there's this process called alternative mRNA splicing, which essentially, as we'll see, results in the phenomenon whereby a single gene can produce a single pre-messenger RNA, but that then can be used to produce multiple different proteins. So that's what we're going to explore next. And as an example, there are multiple examples, and your book probably uses a different example, but let's just use an example, the glucocorticoid receptor protein. So glucocorticoids, these are hormones. They play various roles. They play a role in the stress response and regulating glucose levels and inflammation and so forth. But they're hormones. So recall, hormones work by making their way into target cells. Once in the target cell, they bind to a receptor. So that's shown here in yellow. That complex then makes its way into the nucleus, binds with DNA, and triggers expression of certain genes. So what we're going to focus on is the gene that codes for this steroid receptor, namely the glucocorticoid receptor. And I just want to point out that there are two forms, namely the alpha and the beta form of the glucocorticoid receptor. So let's take a look at what's going on here. So imagine we have the gene. These white lines represent DNA. So the gene for the receptor is being transcribed. So RNA polymerase 2, this red oval, making a piece of pre-mRNA. Then once we have our pre-messenger RNA, our primary transcript here, again, we can identify various regions that represent exons, all of these boxes here, all these different colored boxes, and these regions that represent introns. What happens is that when this gene is modified, I should say this piece of messenger RNA is modified, we remove the introns and we splice together the exons. And notice between the alpha and the beta form, the first 8 exons are the same. However, it's this very last exon, which if we have the alpha form, that incorporates the yellow exon, but the beta form incorporates the red exon. So it's this last exon that differentiates the alpha versus the beta form. So again, you can imagine if you have these two different pieces of messenger RNA, if they are then translated, they're going to give us two slightly different proteins. Again, namely the alpha and the beta form of the glucocorticoid receptor. And actually in this case, it turns out that the beta receptor can actually inhibit the function of the alpha receptor. But again, the main point here is, here's just a simple case where we took just a single gene, produced a single pre-mRNA, but that gave us two different proteins. Turns out there are other genes that can produce multiple, even a dozen or so different proteins. So just a few comments about how the introns and exons are modified. So how are the introns removed and how are the exons spliced together? First of all, let's just identify some key positions that we find within the pre-mRNA. So if you look toward the bottom here, we see a piece of pre-mRNA. All of the yellow region, that's part of an intron, flanked by 2 exons on either side. I just want to point out this first GU, that's conserved. That's very, very common. That identifies the beginning or the 5' end of the intron. Also notice this A right here, that too is conserved. It's very, very common in introns. That's called a branch point, and we'll see why that matters in a second. But toward the far right, the 3' end of the intron, notice this AG. Again, that's highly conserved as well. So this marks the end of the intron. So essentially what's going to happen is we're going to see the whole intron is removed, and this 5' region right here, as we're going to see, will loop around and be linked to this branch point. So we're ultimately going to form this little lasso-like structure, and that's what we'll see in just a minute. So how does this occur? What are the molecules involved in removing the introns and linking together the exons? Well, we just call these accumulation of molecules spliceosomes. So spliceosomes, they consist of a variety of different molecules, in particular small nuclear RNAs and small nuclear ribonucleic proteins, or SNRPs. They all assemble together and again function as a single spliceosome. And so what the spliceosome does, it recognizes the 5' end of the intron, that GU that we mentioned. That's what's happening here. Here's the branch site, there's the A, and it also recognizes the end of the intron, that AG toward the right here. So it then removes that whole intron, links the one side around to the A site, to the branch site, removes the intron, and then links together the exons. So essentially that's the role that spliceosomes play in this whole process. And at that point, that concludes our discussion of RNA transcription and processing.